Now we discuss about the topic that is sphingosine phospholipid. In earlier video we discussed about the glycerol based phospholipid. The difference between sphingosine based phospholipid and a glycerol based phospholipid is, in case of glycerol based phospholipid, there is glycerol present, fatty acyl chain and phosphate group attached with the glycerol, and in case of sphingosine based phospholipid, the fatty acyl chain and the phosphate is attached with the sphingosine, that is also an alcohol. Now the first word which have to discuss when we studied about the sphingosine based phospholipid, that is ceramide. Ceramide is main backbone of sphingosine based phospholipid, and it consists of sphingosine plus one fatty acyl chain. So sphingosine plus one fatty acyl chain, we called it as ceramide. Now if we hydrolyze ceramide in some purpose, so we can get only one fatty acyl chain, but on the other hand, if we hydrolyzed DAG, diacylglycerol, is a phosphoglyceride, we can get two fatty acyl chain. So this is the basic difference between a DAG and a ceramide. So now sphingomyelin. The first point says it consists of phosphocholine head group and sphingosine and a fatty acid chain. So sphingomyelin. So what is when a phosphocholine head group is attached with the ceramide, it called it as sphingomyelin. In case of phosphocholine, that is a glycerol based phospholipid, the head group is also choline. So sphingomyelin is also contain a head group that is choline. So both sphingomyelin and phosphocholine is present outer lip plate of the plasma membrane. Now synthesized in ear. Okay, the myelin sheath rich in sphingomyelin. The myelin sheath which is present in the nerve are rich in sphingomyelin. Not only RBC, if we study about the RBC plasma membrane, we can get that the plasma membrane of the RBC abundant in sphingomyelin. So now how the sphingomyelin is formed, or what is the substrate that form the product sphingomyelin? The phosphatidyl choline and ceramide, with the help of enzyme sphingomyelin synthase, the sphingomyelin is formed. Along with sphingomyelin, there is another glycerol based phospholipid, that diacylglycerol, is formed. So phosphatidyl choline attached with the ceramide, with the help of the enzyme sphingomyelin synthase, sphingomyelin and diacylglycerol is formed. Here the enzyme sphingomyelin synthase, here is the important another term, that is synthetase. There is a difference between synthase and synthetase. In case of synthetase, they used ATP to proceed the reaction, or to proceed the reaction, but in case of synthase, that does not require any type of ATP or energy to complete the reaction. So in case of in the formations of sphingomyelin, the sphingomyelin synthase is involved, and there is no requirement of ATP or. So when we discuss about the sphingosine based phospholipid, a most common word arises, that is lipidraft. What is lipidraft? Microdomain of plasma membrane that contain cholesterol, glycolipids, and here missing the word is the protein. Some glycoprotein is also present in the lipidraft, but major constituent is cholesterol and glycosphingolipid. So we don't know what is glycosphingolipid. We discuss it later video. So glycosphingolipid is if you imagine simple, that glycosphingolipid mean glucose or any type of carbohydrate residue that attached with the sphingolipid, that is ceramide, it called it as a glycosphingolipid. So lipidrafts are involved in signal transduction. So we know what is signal transduction. When the ligand bind with the receptor, there may be some downward pathways, that signal transduction. So the lipidraft takes part in the function, that is signal transduction. endocytosis and cholesterol trafficking. Now, rafts may have cholesterol to sphingolipid ratio of 2:1. The main constituent of lipid raft is glycosphingolipid and cholesterol, the major component. And the cholesterol, the amount of the cholesterol in lipid raft is so much high than the glycosphingolipid. Now the next point, sphingolipid and cholesterol in lipid raft have slightly higher affinity than other lipid molecules of the membrane. So where lipid raft, we can find the lipid raft randomly in the plasma membrane, and inside the lipid raft, the cholesterol and the glycosphingolipid between the affinity between the cholesterol and glycosphingolipid is more, so they form a dense structure than the other phospholipid or sphingolipid. Now we forget to discuss an important sentence or term, that in case of sphingolipid, the fatty acid tail attaches with the sphingosine via amide bond. So, but in case of glycerophospholipid or phospholipid, fatty acyl tail is attached with the glycerol via ester bond. So in case of the sphingosine based phospholipid, the fatty acyl chain attached with the sphingosine with the help of an amide bond. But on the other hand, the fatty acyl tail, in case of glycerophospholipid, the fatty acyl chain attached with the glycerol with a ester bond. If you like the video, please share with your friends. Thank you.